Good afternoon. Thank you all for joining us for this talk and for lunch. My name is Todd Howren. I am the Vice President of RNA Client Services. Over the next 30 minutes, I'd like to introduce you to Aldevron and more specifically talk about some of the complexities, the difficulties, the challenges in bringing an mRNA program to the clinic. And also some of the solutions that we have developed and put together here at Aldevron. So let's jump into it. So I should say, this year is really exciting year for Aldevron. We're celebrating our 25th anniversary. We were founded in 1998, really by two pioneering scientists out of North Dakota State University, Michael Chambers and John Ballantyne, who really saw what was going to become what today we know as the cell and gene therapy market. And so really ahead of the curve back in 1998, and really our history and experience is built off of this genesis that began back in the 90s. So again, founded in 1998, when you look at Aldevron, we have three distinct business units or verticals. DNA, plasmid DNA, which is primarily focused on viral vector constructs, lentiviral and AAV, as well as custom. mRNA, which we will be talking about more specifically today. And then our protein business unit, which manufactures custom proteins with a focus on nucleases, custom proteins, as well as IVT enzymes for mRNA production. Again, we've been in business for 25 years, over 165,000 lots manufactured. Of those, over 400,000 under GMP, supporting phase 1, phase 2, phase 3, as well as commercialization. Geographically, we're located in 3 locations, 3 states. We have a total of 6 campuses, 4 of which you see here. In Fargo, North Dakota is our headquarters. That's where all GMP manufacturing takes place for DNA, RNA, and protein. In Madison, Wisconsin, that's our Protein Center of Excellence, where we do development, process development, design for our proteins. They also manufacture under ISO 13485 for cGMP manufacturing. Those projects are transferred to Fargo for cGMP, tech transferred internally. And then in Lincoln, Nebraska, about a year ago, we made an acquisition of Nature Technologies, which is the home of the nanoplasmid and where we do nanoplasmid retrofit and design, again for GMP production that is transferred to our Fargo-based facility. So really where I want to spend the rest of the time is really talking about mRNA. And what you see here on the slide is for mRNA drug substance production. We began working with clients back in 2015, 2016 on mRNA projects, with the first GMP lot being completed in 2018. To date, we've completed over 70 batches in suite in GMP. And just this year alone, or last year alone, I should say, in 2022, over 250 research grade or demo batches under research grade. All of our GMP batches are supporting phase 1 and phase 2 and phase 3. As well, we do manufacture linear template that supports a commercialized vaccine. So I share this with you because I really want to highlight experience. A lot of CDMOs talk about capacity, which is key. And you shouldn't be in the CDMO business if you do not have capacity. That is what CDMOs are for. But the real differentiator is capabilities and experience. And in the mRNA space, with all the batches that we've done, we have seen all different types of transcripts. We've manufactured all different types of transcripts, whether short or long, GAG capped, ARCA capped, enzymatically capped, encoded poly-A, enzymatically added poly-A, modified nucleotides, saRNA. We have done all of this under GMP, so very broad and vast experience in this area. And that's a real differentiator. Capacity is one thing. Experience and capabilities is ultimately what is going to help make a program successful. So I'm really very excited to announce that we have recently expanded our capabilities in our RNA manufacturing. Last week, we formally announced the launch of our LNP encapsulation and aseptic fill finish services. And really exciting time for us. The genesis for doing this is really linked to our experience in working with clients and different mRNA programs. The difficulty of managing, the complexity of managing a program all the way from cell bank to drug product, all the way from preclinical to commercialization is immense. And we have had clients that have had as many as 4 or 5 different outsourced partners and vendors. So we've had clients that have had a linear template partner, a drug substance partner, an encapsulation and fill partner, and then layered on top of that and across that, analytical testing partners. And the complexity of managing that and the time lost in managing that is immense and enormous. So our mission has been for the past couple years to fill that gap. And when you think about timeline, for all programs in reducing the timeline, it's really, think about seasonal infectious diseases. You can lose months just in hitting your timeline, just in the shipment and the logistics of getting linear template to your drug substance manufacturer, and getting your drug substance to your drug product manufacturer, getting samples sent for analytical testing. So that is our primary goal. This LNP encapsulation and fill finish capabilities have helped to fill that gap. And what we're calling this and what we're getting to is that we're building a true RNA ecosystem, manufacturing ecosystem, and that under one roof, under a single roof, you can go from MCB all the way to drug product under one quality system, under one manufacturing system, all in one program and project management system, thereby reducing the complexities and the timeline in our projects, in your projects. One of the key factors that has enabled us to do this, and do this in best in class, is that we have been able to leverage our sister companies within Danaher. So about a year and a half ago, a little bit over a year and a half ago, we were acquired by Danaher Corporation. So at the bottom of the slide here, you see some of our sister companies that have come alongside us to support this. From the fill finish side, Cytiva or VanRx, we are using the SA25 for our fill finish. And initially, we're moving forward with P&I for our LNP encapsulation. We will be moving to, and we're already making plans and in the works to move to a T-mixer capabilities as well. And then also from the analytical side, we have a host of analytical solutions that we're using from other sister companies. But it's not just the instrumentation that is and the equipment that's of extreme value. It's the direct access to the expertise that these companies bring in an LNP encapsulation as well as fill finish work, aseptic fill finish. So this slide is looking at a workflow, and I need to make sure I stay within my time limit here. This slide is looking at a workflow all the way from construct cloning all the way through drug product. A few things I'd like to point out here. First, on the drug substance, talking about drug substance. What is of tremendous value to us is that we can lean on our 25 years of experience manufacturing plasmid protein. That is the genesis of Aldevran, and for our template, we're able to leverage that expertise inside. Secondly, on the IVT, through our protein BU, we manufacture internally our own IVT enzymes. A lot of manufacturers of IVT enzymes available, but our ability to insource that provides considerable value. Most obviously is supply chain. We have a reliable supply chain of IVT enzymes, but secondly is the IVT enzymes that we use internally. We have developed these, we know how these enzymes function, not only at a small scale, but also at a GMP scale. So when we step in that suite, we can be confident in the results and how those enzymes will perform. And then finally, as I mentioned on the LNP encapsulation and the fill finish, we're leveraging our in-family expertise and knowledge through Cytiva and P&I. So one of the, another big bottleneck that we see is analytical testing. And many times this can cause significant delays in programs. So we have qualified panels, if you will, for both drug substance and drug product, qualified in-house that we are able to utilize for our programs. In addition, we do analytical method development in-house as well as product-specific qualification as needed for some of the assays. The only assay that one of the assays that we do not have in-house right now is cell-based potency assays. We will be bringing those in, but as of today, those are outsourced. So just a high-level view of some of our capabilities, both around research grade, demonstration batches, as well as GMP. Capabilities, capacity to do over 300 research grade or demonstration batches per year. That's at a scale of 2 mil IVT up to a 12 mil IVT. Oftentimes we have clients come in. They may have 2 to 5 different GOIs that they haven't down selected. We'll traditionally begin probably at a 2 mil IVT as they're going through that down selection. As they begin to select, we'll move up to a larger batch and begin to do a demonstration batch at an appropriate size. And this is critically important in doing the demonstration batches, as you want to make sure that when you step into the GMP suite that you have a process, you have a procedure, you have a method that you're confident that's going to be successful. So in our demonstration batches, we use similar equipment as will be used in the, and procedures that will be used as we move into GMP. On the GMP side, again, fermentation capability as well as a linear template, leveraging our background and experience in plasmid DNA production. And then of course, mRNA production. We have 3 suites available for mRNA production, 2 dedicated RNA suites and 1 flex suite, if you will. Not only are the suites dedicated, but the operators are dedicated and trained for RNA. That is their function, that is their purpose, that is what they're trained for. Certainly we have a capacity easily hitting 100 batches per year. What you're seeing here is a picture of our breakthrough campus. This is our headquarters, if you will. in Fargo. This is where we do all of our GMP manufacturing production. In addition to this campus in Fargo, we have 2 other campuses, one for research-grade DNA and a second for research-grade RNA. These campuses are located 2 to 3 miles away from this facility. 2 wings in this campus, north wing, which is at the top of the screen, and then the southern wing, which is at the top of the screen, and the north wing, which is at the bottom left, in between, connected by our warehouse and shipping and receiving. GMP suites in both of these wings. The RNA suites are located in the north wing. So just a little bit on our pilot study for LNP Fill Finish. Very exciting. This was just completed. We're in the middle of our GMP batch right now. This will be completed very soon, but some preliminary data with respect to this pilot study. We did switch. So this pilot study was done with Dasher mRNA. So Dasher is just a GFP Dasher mRNA, and we did this work with the Blaze nanoassembler, which is from P&I. And as you can see from the results, we had a very consistent and very nice size as well as distribution, and then our encapsulation at 97% and very good concentration yield. So very excited about this data. The GMP batch is in flight right now, and we'll have that data soon, and we anticipate initiating our first client batches in the 4th quarter of this year. So I just want to briefly comment on program management. There are instances, very common instances, where our program managers, they're in contact and working in partnership with our client program manager and in partnership with an LNP encapsulation program manager, in concert and connection with an analytical testing program manager. And it becomes a logistics nightmare trying to manage the inner workings of 2 or 3 different partners, trying to arrange slots, trying to make sure those slots line up appropriately. Through this program and through the RNA ecosystem that we're building, from MCB all the way through drug product release, this can be managed by one program manager, streamlining the process, removing the barriers and the logistics of having to make sure all the slot dates line up, they're working in tandem, and also it mitigates that shipping and logistics time. We have seen partners lose a month plus just in the time that it takes to ship an RNA drug substance to the next vendor that's going to do encapsulation and fill finish. First, you have to ship it, especially if it's going to Europe from the United States. You have to deal with customs, and then making sure we release it on a spec and with documents, and then it's the receiving party still has to do their due diligence, their incoming due diligence, and may require extra documentation. So there's quite a bit of time lost just in that handoff. So we can now manage that all under one project management, program management system. Our objective as far as timeline and what I don't want to say for all programs, every program is different, but really to be able to complete a project and go from MCB to drug product all in less than 12 months. So just somewhat on my final slide here, what I encourage everyone to do if you're working with the CDMO, looking for a CDMO, CDMOs often talk about capacity. Capacity is important. It's a chip in the game for a CDMO. But really to ask yourself, does the CDMO really have the capabilities and the experience to help make your program optimal and successful? That's key. We have built this ecosystem, this RNA ecosystem, to enable quick, efficient, streamlined production of RNA drug substance all the way from MCB all the way to the finish. And to really announce and to, first of all, for our 25th year anniversary, and really to kick this program off, we are announcing an early access program. And I would love to have a conversation with anyone that is looking for a CDMO partner. And through this early access program, there will be considerable benefits to our first few clients that will be enrolling and signing up for the manufacture of RNA drug substance or product from Aldevron. So I would very much be happy to have that conversation. Please feel free to reach out to me. And thank you very much, and I'd be happy to take any questions.
Todd, congratulations on the fill finish. But I look around, it still will take almost 1 year to start from the construct to the product. Any kind of space you can minimize?
Yeah. So, absolutely. When you look at streamlining production, it's all a risk-based model. How much risk are you willing to take? The release of the drug substance, let's start with MCBs, right? So you may want to run fast in a program. One of the things we recommend, if you haven't, one of the longest things that can take is just to get your MCBs made, right? So if you haven't completely down selected, maybe you go ahead and make 2 to 5 MCBs, so you have those ready to roll once you've completely down selected. Now, that's a risk-based model that you have to make. When you make your linear template, are you willing to roll into drug substance without the full analytical panel being run? Yeah, there could be some critical parameters that you want to look at. Those could take 1 or 2 days, right? Are you willing to accept that risk-based model? Same way with drug substance to drug product. Risk-based model. Do you need the full analytical panel run, reviewed, qualified, signed off by QA, or are you willing to accept a risk-based model and move forward? So there are ways to streamline. Yeah. And especially in, when you think about the seasonal infectious disease, right? You kind of have to operate that way, right? You're not going to get to market in time if you don't. Good question.
One more question, very quick. So you show us the size, the drug product, after TFF, after the filtration. The size is excellent, 89. So what's the zeta potential? Do you have that? The zeta potential. The zeta potential, the zeta, it's positive, it's negative, the zeta potential, the nanoparticle.
You're asking me a question I don't know offhand. I can get that for you.
I'll email.
Please, yeah, please, thank you.
Do you also hold a DMF?
So,
Or and follow-up is you support, is there a regulatory file support?
yes, we do not have a DMF necessarily for this process, if you will, manufacturing process. Regulatory support, absolutely. We support we can help write sections, CMC sections. We have a service where we will come alongside our clients and help draft. We can even fully write certain sections for our clients.
And do you have clients that's already in phase 3 or GMP?
Yeah. We have a couple clients already in phase 3. Yep. Hopefully move to commercialization soon. Fingers crossed.
Can you elaborate more on the fill finish side, like capacity capabilities and whether you have bio capabilities?
Yeah. So for the fill finish, we are using the SA25. Right now, we feel we will grow with our we have one. We will grow with our customers as they expand. Depends on vial size, vial type, but somewhere around 10,000 vials per day. So we feel we're in a position right now to support phase 1 up to phase 2. And as our clients grow and expand, we will add as necessary to increase that throughput. Starting with 2R and 10R, yeah. And the SA25 has the capabilities to if we need to go to a larger vial size or even syringes, it's modular and we can make the adaptions and will as clients need.
Can you tell us a little bit more about the enzyme you talked about for the IVT? Sounds like you produce your own enzyme. Is that what it is?
We do.
Is there something special about that?
Yeah. So our in-house enzyme right now that we're using is a wild type enzyme. I will share with you, and we use other vendors' enzymes as well. So if a client wants to tech transfer in their process or their enzymes, we do that as well. We will use whatever the client dictates, if you will. I will say we'll probably have some announcements before the year end is out about new T7 polymerases, RNA polymerases, and we have some fantastic data on new mutant enzymes coming to market. So, yeah. But our enzyme that we use in-house is just a wild type.
When it comes to testing and evaluating your RNA, what do you find is the bigger bottleneck time? Is it the actual data acquisition or the data processing?
Data acquisition or data processing? Well, it's going to vary from test to test, right? You know, a lot of times certainly for drug substance, you know, sequencing takes a long time to actually do the work. You know, something like endotoxin testing or sterility is a relatively short test. And it all varies on what we're testing for. Yeah, I know I probably didn't answer your question, but it varies considerably from test to assay, what the assay is. And when you said data collection versus processing, yeah. And I could ask, what do you mean by processing? Is that QA review and release, or?
Whatever you need to do between the time the data is collected and you can say, okay, this product meets all of our specifications.
Yeah, so QA review and release testing. You know, it varies. You know, let's say endotoxin. Endotoxin is a quick, easy test, right? And so you can do it in a day. But the QA review and testing, you know, review may take longer. But that review is a lot of times held up until the complete panel is completely tested. So it's a chicken and egg situation, if you will.
Can you comment about your experience and capability of making longer mRNAs, such as like base editors as well as self-amplifying?
Yeah. So in GMP, up to GMP, we have gone over 12 kb in GMP. Now, in RG, we've gone longer than that. That hasn't progressed to the suite, GMP suite, but in GMP up to 12, right? So we've done we've done a couple programs, saRNA programs, which traditionally have, longer transcripts. So, and then we've gone as short as don't test me on couple hundred bases for IVT on the shorter end. Yeah.
Are there any plans to offer LNP encapsulation of RNPs? Like Riskbased?
Yeah, hey, that is a fabulous question. And the vision that we have is that we will expand the encapsulation to DNA as well as potentially proteins. So that hasn't been worked out, but that is in the vision to complete, yes. Yeah. All right, hey, thank you very much. Please feel free to email me or reach out. Thank you.